Good morning. In the previous video, we were talking about the characteristics of cancer cells. In this video, we are going to continue our discussion of cancer cells and in particular of the role of the inhibitory protein P53 and the proto-oncogenes in the development of cancer. In the last part of the previous video, we said that cancer cannot result from a single mutation, but it results in a cell or a group of cells in which a group of mutations that were accumulating resulted in the occurrence of cancer. And there was a hypothetical vision of the accumulation of mutations in cancer cells or in abnormal cells that were capable of causing cancer. And the hypothesis says that there is usually an initial mutation that will affect essentially a negative cell regulator. A negative cell regulator is a protein that will inhibit the cell cycle. So if the negative cell cycle regulator is itself inactivated by an initial mutation, then the inhibitory proteins in this cell will not be capable of stopping the cell cycle if they detect an abnormality. And by starting by this mutation, the cancer cell makes sure that the inhibitory proteins will not be capable of arresting this cancer cell because they detected an abnormality in its DNA. The next mutation will result in the same effect, but through affecting a positive cell regulator. If you have a protein that acts as a positive cell regulator and you overactivate that protein, then this mutation that includes overactivation of a positive cell regulation will result in encouraging the mutant cell to progress through the cell cycle despite the mutations or abnormalities in its DNA. And then we will have a third mutation that will inactivate a genome stability factor. There is a gene called genome stability factor that is expressed into inhibitory proteins. So if this genome stability factor is itself inactivated, then it will be incapable of inhibiting the cell cycle and arresting the mutant cell to prevent it from division and from propagation. The effect of all these mutations together, this is what we mean by accumulation of mutations. We do not mean a cumulative effect in time, but they're all present at the same time. So the cumulative effect here means the addition effect, the summing effect. All these mutations, when they accumulate in a cell, they cause this cell to be a cancer cell that is capable of escaping the checkpoints of the cell. So as we have just explained, the cancer cells usually display mutations of two types of cell regulators that may result in development of cancer. Number one, mutations in the genes affecting the positive regulators to make them more activated or to make them overactivated. So the first type of mutations that occur in a cell that will be a cancer cell is a mutation in a gene that affects or that is expressed into the positive regulator. And if that gene is mutated, it will make the positive regulators more active. And those genes are therefore considered oncogenes or oncogenic because they will result in a rapid, uncontrollable cell division. The effect of the overactivation and overexpression of the positive regulators is very important. And we have here two examples of positive regulators whose genes become overactive and that become oncogenic, because when you have the gene that codes for the growth factor receptor, and that gene is overactivated, this means what? This means that this receptor, this protein molecule that is the receptor for the growth factor, will not wait until there is actually a growth factor molecule that binds with it to send signals for cell division, but this mutant growth receptor will send signals even when there is no growth factor binding to it. So the gene that codes for that receptor, which is the receptor for growth factor, has been mutated and has become an oncogene because it resulted in a mutation. The other gene is the gene that codes for the proteins cyclins. We have talked about the protein cyclins in the previous video, and we mentioned that cyclins are important for the progression of the cell cycle, and even before progression, the triggering of the reactions that result in progression of the cell cycle. So if cyclin is overexpressed, then it will result in, sometimes it results in inhibition, as we will see in a more deep discussion, but it might also result in a rapid rate of cell division and cancer because the gene that codes for cyclin has been mutated and becomes overactive. So the mutation of a gene does not always result in a gene that is in a protein that is weaker, or that results in a protein that is underexpressed. But sometimes the mutation results in overactivation and overexpression of the gene, as I've just explained. But just like there are mutations that occur in the genes that code for positive cell cycle regulators, there are also mutations that affect the genes that code for the negative regulators of the cell cycle. Of course, the negative regulators of the cell cycle, when they are functioning properly, they are supposed to prevent cancer, because they are negative regulators, meaning that they will stop the cell cycle, they will prevent the cell from pursuing the cell cycle or progressing in the cell cycle if they discover any abnormality. So the negative regulators, the negative cell cycle regulators, are considered suppressors of cancer or tumor suppressors. But if the tumor suppressor itself, that is supposed to detect the abnormal mutated cancerous cell, if this negative regulator itself that is supposed to stop the mad cancerous cell is itself mutated, then nothing will stop the cancerous mutated cells from progressing in the cell cycle. So if the tumor suppressors are inhibited, then those tumor suppressors were supposed to bind with a protein that stops the cell cycle and prevent progression in the cell cycle in response to DNA damage. There are certain sensor proteins, as we will explain very soon, in the cell that bind with DNA and detect damage in DNA, and therefore activate the tumor suppressors and activate, for example, P53. if those sensor proteins that are supposed to be the first detectors of mutations are themselves mutated, so they will no longer sense the damage, and therefore they'll not be able to trigger a response. They'll not be able to activate the P53, which halts or stops the cell cycle. So one of the most famous tumor suppressors, one of the most famous core regulators of the cell cycle, is the tumor suppressor protein 53, which plays a very important role in the cellular response to DNA damage. Of course, the cellular response to DNA damage is to stop its progression in the cell cycle, to stop DNA replication, to stop mitosis, because such a cell that has DNA damage is not allowed to continue interphase and mitosis, and therefore it's not allowed to have daughter cells, because if the parent cell has damage, then this damage would be transferred to the daughter cells. That's why P53 acts very early in the cell cycle, as we have seen in the first diagram on the cell cycle. It acts at the G1 checkpoint, and the G1 checkpoint is present at the end of growth phase number 1, meaning that it occurs before the transition to the synthesis phase, where the cell is going to replicate its DNA. And that makes great sense, because if the checkpoints, if the sensor proteins detect abnormality in the DNA during the growth 1 phase, then this cell is not allowed to enter the S phase. It wouldn't be allowed, of course, to enter the synthesis to reach the synthesis phase and replicate its DNA. And that's why we call the G1 checkpoint the restriction point. And it's a very important checkpoint, because if any DNA damage is detected here, the cell is not allowed to enter the S phase, and the cell is arrested at the G0 state or G0 state. At first, as we said in the previous video, temporarily, to see whether the DNA damage is repairable or not. And if it is not repairable, the arrest in G0 would be permanent. As I've just said, and here we are going to see the diagram, that there is a sensor protein that detects any damage in DNA if present, and in response will activate the inhibitory protein P53. And of course, the response of P53 is the immediate stopping or halting of the cell cycle by synthesizing an inhibitor. So here we have 3 important proteins. We have the sensor that detected the damage in the DNA. We have the second protein, which is the inhibitory protein P53, and we have the third protein whose production is triggered by P53, which is a cell cycle inhibitor. So in this diagram, we see the normal condition. When we have a normally functioning P53, a non-mutated P53, because what is really incredible and amazing is that the P53 itself, that is supposed to respond to mutations in DNA, is or might itself be mutated. So here we are dealing with a condition in which we have DNA damage. And the DNA damage, in case the P53 is normal, then this DNA damage will be detected by a sensor protein, and that DNA damage that is detected by a sensor protein will result in the sensor protein activating P53, but the sensor protein does not show here on the diagram. When P53 is activated, it will bind to DNA. So the first binding was DNA with the sensor protein. And then the sensor protein activates P53. And then P53 binds again with the DNA. Or DNA binds again with P53, which is the second molecule that is capable of binding with the damaged DNA. And the response is as follows. As you can see here, this part of the diagram shows you how P53 accomplishes its task. We have here the cyclin that acts at the junction or at the end of the first growth phase and the beginning of the synthesis phase. That's to say the first restriction point, and that is supposed to allow cell or trigger cell cycle progression. But we also have the CDK, cyclin-dependent kinase, because the cyclin alone cannot work without the cyclin-dependent kinase. They both depend on each other, and the kinase is responsible for phosphorylation of certain proteins to allow for cell cycle progression. But as you can see, when the P53 is activated and when it binds to the damaged area of DNA, you can find that this there is a molecule here that you see baby blue in color, and that is called CDK inhibitor. This is the inhibitor that we have just talked about when we said that P53 will trigger production of a cell cycle inhibitor. So this cell cycle inhibitor does what? This cell cycle inhibitor binds to this complex, cyclin CDK complex, and prevents cell cycle progression. So when there is prevention of cell cycle progression, this is the ultimate result. But how does it occur in detail? First of all, the cell is halted or stopped at the G1 checkpoint. This pause will buy time for repair of DNA. So the P53 molecule is giving a chance for the DNA of the cell to be repaired and for the repair mechanisms to be activated. And if the repair, if the damage is extensive, and if the repair fails, then there will be triggering of apoptosis, and the arrest in G node will be permanent if the repair is impossible. And the purpose of all this is not to allow the cells with damaged DNA to divide and pass on their abnormal genetic traits to their daughter cells. But in case the P53 itself is mutated, even when the sensor protein senses the presence of DNA damage, P53 cannot be activated, which means that the sensor protein tries to activate P53, but because P53 is mutated, it does not respond. And because it did not respond, or it couldn't respond, we didn't see here the baby blue molecule, which was the inhibitor of this complex whose synthesis was activated by P53. So no activation of P53, no binding of the mutant P53 to DNA, no formation of the inhibitory molecule, and therefore the cyclin CDK complex stays active and is not activated, and therefore the damaged DNA is passed on, although there is a damage that we cannot repair because the cell here, the cell here is supposed to stop at the G1 checkpoint to repair the damage. Here, if you see in the above diagram, we see that the cell stops at the G1 checkpoint and the DNA repair is activated, the repair mechanism is activated, and if the repair is impossible, then apoptosis is triggered. But here, in the second part of the diagram, in the second part of the diagram that I've just skipped, I'm sorry, I thought I was pausing the recording, but I wasn't. So in this second part of the diagram, everything that was supposed to occur, like halting at the G1 checkpoint, like repair, activation of the repair mechanisms, like triggering apoptosis, all this does not occur. And the mutant cell is allowed to continue and to progress in the cell cycle, and therefore it is allowed to transfer its mutant DNA to its daughter cells. So again, as I've just said, the pause in time buys time, the pause in the cell cycle buys time for DNA repair. And this is the function of P53. So the second job of P53 is after binding with the damaged DNA is to activate the DNA repair enzymes as we have just explained. And then if the damage is fixed, P53 will release the cell. It will receive a go ahead signal. Just continue your journey in the cell cycle and it will end up by giving daughter cells. But if the damage is not fixable, then P53 will play the third role, which is triggering apoptosis. So again, if the P53 molecule detects a damage, it stops the cell cycle temporarily to try to fix that damage. If the damage is fixed, then the cell is allowed to rejoin the cell cycle. It receives a go-ahead signal. But if it is not fixable, and if P53 is doing its function perfectly, then it will trigger apoptosis. It will make a decision, a very important decision, that this cell should undergo suicide, which is the programmed cell death. It's a type of very organized cell death to prevent the damaged DNA from being passed on to the daughter cells. Another characteristic of the cancer cells, as I said in the previous video, is that the cancer cells can invade other tissues. Unlike the benign tumors that are non-malignant and that are well encapsulated, the presence of a smooth, well-formed capsule around a tumor or a mass of cells is a good indicator that this is most probably a benign tumor. But if you have a mass of cells that is formed of cancerous cells that have the ability to secrete enzymes to digest any capsule formed and to even digest and invade the normal cells around the tumor, then if you see the the tumor branching like this and having a fuzzy outline, an irregular outline without a well-formed capsule. And if you find that it is richly supplied with blood and highly vascular, this indicates that it's a highly active malignant tumor. So this table is very useful to show you the very important or striking differences between the benign tumors and the malignant tumors. The benign tumors are made of normal cells that are not mutated, but so their growth speed is slow, whereas the growth speed in malignant tumors is very fast. As we said, the benign tumors have a well-defined regular capsule, whereas the malignant tumors have a poorly defined irregular capsule because they are trying to invade the other tissues, the neighboring tissues, with the help of enzymes they secrete. And since the benign tumor is well encapsulated and does not erode that capsule or invade the surrounding tissues, then the benign tumor is localized and non-invasive, whereas the malignant tumor is invasive and can spread to other tissues. That's why when you remove the benign tumor surgically, it's rarely recurrent, but of course, there is a high rate of recurrence in malignant tumors, and therefore there is no metastasis or spread to distant areas in case of the benign tumors, whereas there is often metastasis via blood or via lymph or even direct metastasis by invasion of the blood vessels or invasion of the lymphatic tissues or invasion of the lymphatic vessels, or invasion of the surrounding tissues. In the last part of the video, we are going to talk about the oncogenes. The oncogenes are normal genes, but the oncogenes are the genes that are capable of causing cancer, but they are derived from normal genes. So I repeat again that the normal genes are called proto-oncogenes, and these proto-oncogenes are liable to be converted into oncogenes. So what's the relation between the proto-oncogenes, the oncogenes, and the cell cycle regulators? As we said before, we might see in some cancerous cells that a growth factor receptor is overactivated because of a mutation in the gene, and a cyclin may be overactivated because of a mutation in the gene that is expressed into cyclin. This is related to our topic that we are explaining now, which is proto-oncogenes. What are the proto-oncogenes? The proto-oncogenes are normal genes, and these genes are supposed to be expressed into or are supposed to code for the cell cycle regulators that regulate the rate of cell growth, the rate of cell division, and the lifespan of the cell. But we know that the cell cycle regulators might be positive regulators or negative regulators. Suppose we are taking the example of the positive regulators, like the growth factor, for example, or the cyclin. So the normal genes that code for the growth factor receptor or for cyclin are called proto-oncogenes. But when they are mutated and when they result in overactivity of the growth factor receptor or of the cyclin, then they are oncogenes because they will result in mutation. So the normal genes are called proto-oncogenes, proto, because they are liable to give oncogenes if they are mutated or if they are overexpressed, and therefore they become cancer-causing genes or oncogenes. So what are oncogenes? The oncogenes are the mutant forms of the proto-oncogenes, or the oncogenes are the cancer-promoting forms of the proto-oncogenes. The overactive cancer-promoting forms of these genes are called oncogenes, while the normal non-mutated form of the genes is called a proto-oncogene. And the naming system reflects the ability of the gene to cause cancer or not. So an oncogene is the mutated one because it will cause cancer. And the proto-oncogene is a normal one, but that is liable to be mutated, in which case it would have an overactivity, or it would be overexpressed and would result in cancer. We have different types of mutations that can affect the proto-oncogenes and make them be converted into oncogenes. The first mutation is a mutation that changes the amino acid sequence inside those proteins, because the cell cycle regulators are proteins that are made of amino acids, and the amino acids are translated out of the genes. So if the gene is mutated, then the amino acid sequence will differ, and the final protein, whether cyclin or growth factor regulator, will also be abnormal. So there are some mutations that result in changes or alterations in the protein in such a way that the shape of the protein is changed, and this change in conformation of the protein, like the growth factor receptor, makes it always in the on state. It never becomes in or gets into the off state. Another mutation will not affect the conformation of the protein and would not result in the cell cycle regulator being always on, but it might involve an amplification, meaning that there will be extra copies of the gene, and therefore the gene will start making too many copies of the protein. So the second type of mutation is the mutation in which there is amplification. The first one was permanently turned on. The second one is amplification. So you have a gene that codes, for example, for the growth factor receptor that binds with the growth factor to initiate cell division, and that gene becomes amplified. What does this mean? This means that the cell will have extra copies of the gene. And if we have extra copies of the gene, then those copies will be translated into much, much more proteins of positive cell cycle regulation. And therefore, the cell cycle will occur rapidly, and there will be a mass of abnormal cells called cancer cells. Sometimes the third mutation is an error in repairing DNA, meaning that there is an error that occurs during the process of replication in which the normal gene, proto-oncogene, is attached to a different gene during replication without separation between them, without, for example, the presence of the promoter and the initiating sequences that denote the beginning of the gene. And therefore, we will have a bigger gene, a proto-oncogene that was erroneously or by mistake attached to a neighboring sequence, resulting in a bigger gene that we call a combo gene or a combo protein. And needless to explain, of course, or to say that this combo protein will be translated into an abnormal protein. We will see here in the following diagram an example of how the gene mutations might result in abnormal cell division. We will take the example of a famous protein that is called the RAS protein. The RAS protein is a G-coupled protein, as we explained previously in the unit on the cell, and the video is on the channel. The G proteins are cell membrane proteins that are bonded to GDP or to GTP, guanosine diphosphate. It's a nucleotide, it's a dinucleotide, or guanosine triphosphate. If they are bonded to GDP, then they are in their inactive state. And if they are bonded to GTP, they are in their active state. And the normal is that this RAS protein is a receptor. It's a receptor on the cell membrane, and it is activated only when the signaling molecule, specific signaling molecule, is attached to it, and in which case it becomes activated and bonded to GTP and becomes on. But if there is, this is the norm, but if there is, we will see, of course, the details on the diagram, because it's hard to understand without the diagram. If we have a mutation that will cause cancer, this means that this receptor protein, which is called the RAS protein, has been affected by that mutation in a way that makes it no longer switch to its inactive form. It will always be in the on state. It will always be in the active state, and therefore it will always activate the process of mitosis. So the mutation makes it no longer switch to its inactive form, or switch to its inactive form, but very slowly. And we will see here how this occurs. So in the normal condition, we have the RAS on the cell membrane, which is a receptor on the cell membrane, and we have the, or actually the RAS is a part of this structure, which is the growth factor receptor, and it is a protein that will be activated by that receptor, but it's also on the cell membrane. So we have here the growth factor receptor, and if you have molecules of growth factor around the cell membrane or close to the cell membrane, the growth factor is the specific signaling molecule to the growth factor receptor, and the growth factor receptor is connected to this G protein, which is the RAS receptor or the RAS protein. The RAS protein will be activated and will bind to GTP, and this will result in cell division and proliferation. And in the normal condition, we also have another possibility. The first possibility, so these two possibilities are for normal RAS, when you have a normal G-coupled protein called RAS. But here we have the normal RAS in the presence of the growth factor, and here we have it in the absence of the growth factor. The normal is that when there are molecules of growth factor, it gets stimulated and bonded to GTP and starts cell division and proliferation. But if there are no molecules of growth factor in the vicinity, then the receptors for growth factor will not receive their specific signaling molecule. So RAS is not activated, so it remains bonded to guanosine diphosphate, which is the inactive status of RAS, and therefore RAS stays in the off condition, and there is no cell division or proliferation. These two conditions I've just explained are the norm. But if we have a mutant RAS, if we have a mutant RAS, or if we have an oncogenic RAS, you can find that even when there is no growth factor in the vicinity of the cell, in the surrounding fluid around the cell, even when there is no growth factor, where there are no growth factor molecules, the RAS stays bonded to GTP. The normal is that when there is growth factor, it becomes dissociated from GTP and becomes bonded to GDP. But here it stays on because it stays bonded to GTP, and there is continuous cell division and cell proliferation, which results in cancer. I hope this video was useful, and if you found it useful, please click on like, send your comments, share it with your colleagues, and subscribe to the channel if you haven't already. Thank you, and see you on the next video.